Hello there. My name's Dan, biology tutor. This is going to be a short video on the AS topic, cell physiology, specifically the topic osmosis, which is about water potential and solute potential and those sorts of things. Just before I start, you'll see my details on the screen there, my mobile number, and I do tuition for GCSE biology, GCSE physics, AS biology, and A2 biology. Now, if you're doing tuition with me, you should have already received a package of materials which I'll post to you. So if you get in contact with me by my mobile number there, just text me and say you want to do a particular topic, whether it's any of those areas at all. What I do is I post you a full package of materials that you can use then whilst you're watching these videos or while you're Skyping with me. Now, get a successful tuition session, you really need these materials. So, for example, for this topic, osmosis, an AS topic, I would supply you a full set of my notes, which are very tight and structured and crafted so that they contain all the most important bits and pieces, but with some of the excess material that's sometimes present in school notes trimmed out, so really just the essential information. And they're very good notes, they're sought after. So if you get in touch with me, I can send you a set of notes for a topic. I'll also send you a set of questions, a question booklet for the topic with questions that are crafted to cover the topic perfectly. Also a set of answers for the questions for that topic. And also other various materials, including these short questions, which are designed to really test your ability to understand the topic once you have finished your preparation and you feel you know the topic well. So what you want to do in the first instance is get in touch with me by text, and I'll send you a package of materials which you then can use whilst you're watching this video or whilst I'm Skyping with you, whatever you choose to do. So as I go through this video is going to be on the topic osmosis. It's part of the AS topic, cell physiology. Probably by this stage you've already done topics in AS biology, probably done cells and cell ultrastructure, and possibly done molecules as well. And this then would be a follow-on from those topics. So as I say, you need to have my notes in front of you, really here, to get full benefit from this video, because I've been making constant references to the notes, to certain pages, to diagrams, to photographs, etc. I'll also be making reference occasionally to questions as well, and going through questions. And so you need the whole thing in front of you. If you have everything in front of you, then you can follow what I'm doing here. Okay. So. Let's get underway then. So this topic, cell physiology, consists of essentially two parts. Cell physiology has the first, it needn't necessarily be the first half, but one half of it is on the topic osmosis, which is about water potential and solute potential, and those sorts of things, which you'll recognize if you've done this in class. The other half of this topic then is about diffusion, facilitated diffusion, active transport, and endocytosis and exocytosis. Both are equally important. Both generate questions in past papers, and so you should know both of them. But I think in this video, I'll really only be able to get through the osmosis, the water potential end of things. It'll possibly take me almost an hour to get through that to do it properly, and I want to do it properly. And hopefully at the end of it, you have a better understanding of water potential and so on. This is quite a difficult topic. It's difficult for AS students, and I often find sometimes that some A2 students still haven't got this topic. At A2 you might use water potential when you're doing the topic kidney. And some students will admit to me freely that they never quite got the water potential stuff, the osmosis stuff from AS. So you can, but if you go through this with me, hopefully it becomes a bit clearer. Okay. So, cell physiology, where I'm going to start then. All right, so let's start with GCSE. And let's remember at GCSE that we would have had experiments and discussed what happens if you put, for example, animal cells, for example, red blood cells into water, or what happens if you put plant cells, such as pieces of potato, into water. And you remember those experiments at GCSE. Maybe you didn't do them, where you cut up pieces of potato and put them into water in various solutions, and you observed what happened. So essentially, that's what we're looking at in this topic, except we're looking at it in more detail and with more complexity, and using AS terms rather than GCSE terms. So, as you see, I keep referring to my notes here, and the reason I do refer to my notes is that many of the diagrams and features in my notes are just in the notes, and I won't be drawing a lot of things on the board here because I couldn't get through the topic in even an hour if I start transferring diagrams from the notes to the board. So you need the notes in front of you, and then it becomes easy. You can look down. You look down the notes, and I can point at a diagram in the notes, and you can find it on page 11, and then we can work from that. And the notes are written very succinctly and in nice language, so that hopefully it's understandable and it's well sequenced. So if you go back to GCSE and you're talking about osmosis, you remember there's a definition of osmosis. Osmosis is the movement of water through a semi-permeable membrane, or a selectively permeable membrane, or a differentially permeable membrane. Call it what you like. Most schools would have said semi-permeable membrane. So the movement of water through a semi-permeable membrane from a high concentration of water to a low concentration of water, or from where water is at a high concentration to where water is at a low concentration. Other ways you could have said that is from a more dilute solution to a more dilute area to a less dilute area. In other words, where there's more water to less water. So if you remember that definition, that was the GCSE definition, and you're using words like concentration through a semi-permeable membrane. Now, if you go to AS, when you come to AS level, you get a slightly different version of the definition for osmosis, and you'll see there on page 9, top of the page. Osmosis is the movement of water through or diffusion of water through across a semi-permeable membrane from a high water potential to a low water potential. So instead of saying from a high water concentration to a low water concentration, we're now using the word water potential instead. By the way, I'm going to talk very fast here and go quite fast, because it's going to take me practically an hour to get through this topic, and so I need to move with great speed. But if you have the notes in front of you, it's not a problem keeping up with me, because I'm looking down at the notes here and I'll refer to the notes, and you can look at them as well. So get in touch with me by text if you need the note package, and then you can get the note package and everything in front of you, and then you can go through watch the video. So we're faced with a new term at AS, then we're faced with this term, water potential. So the water potential, rather than the term water concentration. Now, this is complex, and there are a wee bit of maths involved in it, and it's quite difficult. So you'll have to bear with me as I'm trying to explain it to you. There's a definition for water potential there. It's the tendency, or measure of the tendency of water to enter or leave a solution or a cell. So the tendency of water to enter or leave a solution or a cell. Now, there's a question then in the notes that says, how is the water potential calculated? How do you get the water potential of something, of a solution or of a cell? Well, actually, you'll be presented with three different situations where you might be calculating the water potential. You might be discussing the water potential, say, just of a container of water in a container. So you've got water in this bottle here. You might say, what's the water potential of that water? Or you might add something to the water, like some sugar or salt or some other ions or something that dissolves in the water, and then ask, how does that affect the water potential of the water? And so that's the first thing we're going to look at. We're going to look at the water potential of water, liquids, and solutions in beakers and containers. Okay. The second place for water potentials, you'll have to discuss water potential, is animal cells. We're talking about cells like our own body cells, like skin cells. Well, the example that's usually used, or the animal cell that's usually considered in exam questions, is red blood cells. Red blood cells are a good one to use. So, for example, you could ask a question: what would happen if you put red blood cells into water? What effect would it have on them? And so we'll look at that in a moment or two. And the other kind of cells that we need to look at are plant cells. What happens to plant cells when they're placed in water and various solutions? And you kind of remember this from GCSE, because this is what it was about. You spend a lot of time cutting up pieces of potato with cork borers and things, and putting them into various solutions and observing what happened. And you've seen that in some cases water moved into the potato cells, and sometimes it moved out. So that's where we're at. That's where we're looking at. So, asking the question then, how is this thing called water potential calculated? So we're going to look at that. Now, when we're doing the calculation of water potential for either solutions or animal cells or plant cells, there's a formula that's very important, and there's some symbols here that are important, and are difficult and important as well. Now, the basic formula you have is the water potential. Now, water potential is written with this kind of like fork affair. Water potential, that's the water potential, is equal to something called the solute potential plus something else called the pressure potential. So you've got these odd symbols. Water potential is equal to solute potential plus pressure potential. Now, I'm not going to spend an age writing stuff onto the board, because, as I say, this is all read out in great clarity in the notes. So you need the notes in front of you to be able to work on it. Now, when we're working, we're going to look at three situations then. So we're going to look first of all at solutions in beakers and water and so on and so forth. When you're looking at solutions and water and so on, the formula that you need is simply this. You simply need to know that the water potential is equal to something called the solute potential. So that's the formula. The water potential is equal to the solute potential. Okay, so what's this thing called solute potential? So, and how do you get it? Well, there's some rules about this. First of all, the solute potential of pure water, and I'm saying that this here is pure water. Okay, we'll just have to go with that. It mightn't be totally pure. But pure water, or chemists would call it deionized water or distilled water, pure water, the solute potential of pure water is zero. It's zero. So that's the first fact you have to get. Pure water has a solute potential of zero. So this figure here for pure water is zero. So if I ask you then, if the solute potential of pure water is zero, what's the water potential of pure water? And the answer is, it's zero. In other words, if you know the solute potential of a solution or water or whatever, then you know its water potential. So the solute potential of pure water is zero, and the water potential therefore is zero. Okay? So that's the first thing you want to get. Water potential of pure water is zero. Now, what happens if you add some salt or some sugar into this water? Then it's no longer pure water, it's now a solution. You could add sugar, salt, ions, or whatever, something that's dissolved into the water. Then what effect does that have on these values? Well, as soon as you add some salt or sugar or any ions into this water, then something happens to the solute potential. For example, the solute potential might now become minus 100. Okay, and there's units for that, they're called kilopascals, not too important, kPa. So the solute potential is now minus 100. So if we say the solute potential is minus 100, what's the water potential? The answer is, it's minus 100. So it's kind of simple for water and solutions and so on. If you know the solute potential, then you also know the water potential. So adding salt or sugar or anything into water causes the solute potential to go negative. So the highest solute potential you can have is zero. That's for pure water. If you add anything into the water, solute potential goes negative, minus 100. What happens if you add more sugar or salt or any other dissolved ion into this water? Then it might go, this becomes more negative. So it may become minus 200. Okay? So adding more salt or sugar to water or any other kind of dissolved stuff to the water causes the solute potential to lower. You see it's been lowered because it's going from zero for pure water to minus 100 to minus 200 and so on. And the more salt and sugar you add, then the more negative this becomes. And of course, all the time, the water potential is the same as the solute potential. So if you knew, for example, the solute potential is minus 500, then that's the water potential. And this is a rule for liquids and solutions. Notice we haven't talked about cells as yet at all. Okay, so there was a question, once an exam question, it said something like, suggest what you would add to pure water to lower its solute potential, and the answer was solute, some sort of dissolved stuff, which seemed counterintuitive because you thought if you add solute to water, that has to raise the solute potential, but no, actually it lowers the solute potential. So the solute potential maximum value is 0, and it can only ever be less than that if you add stuff to the water, you add ions and so on. So that's the rule for water and solutions, and you can see that all in the notes here. Pure water has a solute potential of 0, and so its water potential is also 0. Now, if you have say some salty water, then let's say you had some water with a lot of salt in it, and let's say its solute potential was, let's say, minus 500. Okay, so its water potential is minus 500. What would happen if you added more fresh water to it? If you added in more pure water, in other words, if you diluted it? Well, the answer is you'd make the solute potential go back this way. So instead of being minus 500, it might be minus 400. So adding pure water to a solution makes the solute potential go more towards 0. Okay, it'll never reach 0 because there's always some dissolved solute. Okay, so adding water to a solution makes the solute potential go more towards 0, and therefore the water potential also goes towards 0. So I'm taking track of my notes here and keeping an eye on them because I don't want to miss out any points, and sometimes if I just talk on, I miss out a point, and then if I come back to it, then it gets kind of awkward and gets in the wrong place. So what kinds of solutions are we thinking about here? Well, for example, seawater is a good example. Seawater is not pure water. It's salty. It's got a lot of salt in it. So it's going to have a solute potential, maybe, I think I've chosen a value. I've said it might have seawater there, might have a solute potential of minus 2,000. All right, not minus 100, minus 200, not minus 1,000, minus 2,000. In other words, it's very salty. It depends on how salty it is. If seawater then gets diluted, sometimes you get seawater that's diluted by river water coming into it. Its solute potential mightn't be minus 2,000, might be minus 1,000. But that's the general principle. Seawater would have quite a negative solute potential, maybe minus 2,000, and that means the water potential of seawater is minus 2,000. I've just said here in the notes, in class experiments in school, and if you're making up experiments, so you remember the experiments at GCSE where you put pieces of potato into different solutions, where you put a piece of potato into pure water, then you put it into weak sugar solution and stronger sugar solution, and a whole range of sugar solutions. Then you could make those up in class, and each of those would have a lower water potential. So the pure water be 0, weak sugar solution might be minus 200, stronger sugar solution might be minus 400, and very strong sugar solution minus 600, and so on. So you can make a whole range of solutions. Now, so addition of solute or any dissolved substance to pure water causes the solute potential or the water potential to go down. In other words, if you go back to the definition of water potential, it reduces the tendency. If you add salt to this water, it reduces the tendency of water molecules to be able to leave the solution, get out of the solution. And the question sometimes asked, as on page 11 there, why does the addition of solute to water cause its water potential to decrease, i.e. become negative? And you'll see I have a diagram there on page 11, and if you have the notes you can look perfectly at the diagram, and I'm not putting the diagram up on the board, it's just it takes too long, and we wouldn't get too far on this topic. So on page 11 you can see the fact there, you have pure water. If you have pure water on this side, and then you add in some sugar solution, add in some sugar molecules there, you can see what happens. I've said there, in pure water, all the molecules in the pure water are free to leave the liquid or the solution. But if you add in sugar molecules into the solution, each sugar molecule acts as a wee nucleus and attracts lots of water molecules to the sugar molecules. In other words, to the sugar molecules. In other words, the water molecules in the solution are attracted to the sugar molecules And the water molecules form like a wee layer around the sugar molecules, and these wee layers are called hydration shells. And these hydration shells then are like just wrappers made up of water molecules all clinging to the sugar molecules. And because these water molecules are clinging to the sugar molecules, these water molecules are no longer available and they're no longer able to leave the solution. So the addition of sugar, as it were, traps water molecules in the solution and reduces its water potential. The more sugar molecules you add, the more the water molecules are held in the solution. I've just said here, addition of solute has reduced the tendency of water molecules to leave the solution. It has reduced the water potential. The more solute that's added, the lower becomes the solute potential, i.e. the lower becomes the more negative or lower becomes the water potential of the solution. Now, other terms that you'll come across here would be the terms hypotonic, hypertonic, and so on. Hypotonic, hypertonic, and so on. Now, basically, those are terms that are used to compare solutions. So, for example, if we had pure water, this is pure water here, pure water in that beaker, and in this beaker you have seawater. So this is seawater, and this is fresh water or pure water. Well, these terms are used, they're not used that frequently anymore in exam questions. but the terms that are used are hypertonic, hypotonic, and isotonic. Now, let's take the term hypertonic. Hypertonic just means a higher concentration of dissolved substances present. Higher concentration. So, for example, we'd say the seawater is hypertonic to the pure water. So hypertonic actually means, so if you looked at their water potential, the water potential of seawater, what we said was minus 2,000. It's minus 2,000. Pure water, the water potential would be 0. So we say that the seawater is hypertonic to the pure water. So hypertonic just means has a lower water potential than. Okay. Now we also use another word, hypotonic, and we'd say the water is hypotonic to the seawater, and hypotonic simply means that solution has got a higher water potential than the other solution. So it's a term of comparison. So for example, I would say, that pure water and a soft, let's say a soft drink, like say Lucozade or something, you would say pure water is hypotonic to the Lucozade, and the Lucozade is hypertonic to the pure water. Okay, so hypertonic means a lower water potential, hypotonic means a higher water potential. Now the other term you come across is isotonic. Isotonic would be the term you'd use for two solutions that have the same water potential. So for example, you might say seawater is isotonic to possibly some solution that you've made up in class. So you make up a solution full of salt and sugar and stuff, and that might be isotonic to seawater. So it simply means they have the same water potential. So those terms that are not used that much, but it's taught in class, so you kind of have to be familiar with them. And it's all dealt with perfectly here in the notes. So if you have the notes, you'll see. Hypertonic, a more concentrated solution than. Now it's a more negative water potential. Isotonic, the same concentration of solutions. Two solutions are isotonic, same concentration, i.e. the same water potentials. Hypotonic means refers to, for something like pure water, it's hypotonic to everything else because it's got a higher water potential than everything else. Okay. Now, okay. Once again, I apologize for going so fast, but I need to work fast. There's a lot of information here, and if you have the notes in front of you, then you can see all the things that I'm saying here are all spelled out very clearly in the notes. So the next place we have to go, we've dealt with solutions. Now we have to deal with animal cells and plant cells. So animal cells and plant cells. Okay. And first of all, we're going to deal with animal cells. So animal cells, and the example we're going to use a lot here is red blood cells. Red blood cells are good because you can do experiments with them. Not so much nowadays, but when I was at school, the teachers were able to take some blood from the students and take a drop of blood from your finger and put it into water, and we could look and see what happens to the red blood cells as well. Not allowed to do that with students anymore, but that's the basic idea. And you'll often get questions about that. What happens if you put red blood cells into pure water? Okay, what happens if you put red blood cells into a strong sugar solution? Okay, you might remember this from GCSE. It's pretty straightforward from GCSE. Now, when we're dealing with animal cells or red blood cells, then the formula, we're talking about how you get the water potential, say, of a red blood cell. Well, it's exactly the same formula as we used before. So the water potential for a red blood cell is equal to its solute potential. So it's the same as solutions. You might think, oh, this is a bit confusing, but that's essentially, so if you know the solute potential of a cell, then you know its water potential. Okay, and you have to get familiar with these terms. So it's the same. So and here's the starting point. You sort of say, well, if you take a red blood cell like any other animal cell, like a skin cell or a liver cell or any other cell, or even a little single-celled organism that's living in a pond water, if you look inside that organism, you'll find it's cytoplasm. If you look inside the cell, it's got cytoplasm. In the case of the red blood cell, the cytoplasm is water with lots of hemoglobin. So it's not pure water inside the red blood cell. It's definitely some sort of solution. It's got water and dissolved hemoglobin. So it makes sense then that a red blood cell doesn't have so its solute potential of a red blood cell is definitely not zero because it's not pure water. The cytoplasm of a red blood cell is not pure water, so it's definitely not zero. So it's possibly some minus value. And I'm just looking at examples that are given in exams here. So they've said here, for example, that the solute potential of a red blood cell is minus 300. Yeah, that sounds good, minus 300. So its solute potential is minus 300 because it's a sort of weak solution inside it, and therefore its water potential is minus 300. Okay, so the water potential of a red blood cell is minus 300. Now, here's where all this water potential and solute potential comes into its own, because now you can ask questions like, what happens if you put a red blood cell into pure water? And explain what happens. You see, pure water, beaker of pure water. There we are. Red blood cell. Not sure about it. A red blood cell. Pop your red blood cell. I'll draw a red blood cell sort of like that there, so you can see that. That's your red blood cell. Then you put it into pure water. Now you want to ask what's happening here, and you want to explain it in terms of water potential. So let's first of all, if this is pure water, then its water potential is 0. So water potential of pure water is 0. Okay. Water potential of the red blood cell, well, I've just said it might be minus 300. So this one, the red blood cell might be minus 300. Minus 300. We're not writing the units here a lot. The units are kilopascals, kPa, not too important. So the question is, what happens if you put a red blood cell into pure water? Well, the rule is that water will move from a high water potential to a low water potential through a semi-permeable cell membrane, through a semi-permeable membrane. I was going to ask you, what's the semi-permeable membrane in this case? And the answer is it's the membrane of the red blood cell. So you can't see it, and you maybe haven't done enough of this yet, but you might know that I'm just rubbing out wee bits here to show that the cell membrane of the red blood cell is full of holes. Little pores. And the pores are big enough to let water through, but won't be big enough to let the hemoglobin through. So only water can move. So the question is, what happens here? The answer is water moves into the red blood cell. Why does water move into the red blood cell? Because water moves from a higher water potential, which is 0, that's the highest possible water potential, to a lower water potential, which is minus 300. Water moves into the red blood cell. As water moves into the red blood, so you're answering this question, say water moves into the red blood cell by osmosis from a higher water potential to a lower water potential through the semi-permeable cell membrane. What effect does it have on the red blood cell? The red blood cell starts to expand, gets heavier, it expands, starts to swell, and eventually it bursts. Red blood cell bursts. And there's a word for this when the red blood cell bursts, and the word is lysis, Lysis, or hemolysis, more correctly, Hemolysis, the bursting of red blood cells. You put them into into pure water. So red blood cells will burst. Put them into pure water, and that's a commonly asked question. Now, the other question you may get asked is, what if you put a red blood cell into not pure water, but a strong sugar solution or a strong salt solution? So this is your strong salt solution. Yeah. What happens if you put the red blood cell into a strong salt solution? Well, the water potential of a strong salt solution might be something like -1,000. Yeah. So the water potential of the solution is -1,000. The water potential of the red blood cell is -300. Which is the higher water potential? I'll show this in here. Water potential of the red blood cell is higher. Water will move from a higher water potential to a lower water potential. So what happens in this case? Water moves out of the red blood cell into the solution, and it keeps moving out of the red blood cell. Red blood cell starts to shrivel up and becomes sort of shrunk, and it ends up sort of looking like this. It shrivels up and becomes shrunk, and it loses lots of water. And that word, there's a word for this here, it's crenation. The red blood cells are crenated, undergo crenation. So that's a common question. It's not usually given a lot of marks because it's considered quite easy. So that's what happens to red blood cells when you place them in a strong sugar solution or place them in pure water. Now, I've asked a question, page 13 there, top page 13. So why is it important that blood plasma is isotonic to red blood cells? Isotonic means the same water potential. Why is it important that your blood plasma has the same essential concentration as your red blood cells? Well, the answer is you don't want your red blood cells floating about in blood plasma, because that's where they're at. They're suspended in the plasma in your blood. You don't want them in plasma that's got a different water potential. You don't want water moving into the red blood cells and them bursting, or you don't want them shrinking. You want the solution that surrounds the red blood cells, i.e. the plasma, to the same water potential. So you'd say it's isotonic. It's isotonic to the red blood cells. Okay, so that question's dealt with there. That question's dealt with in the notes. Note, I'm not going through the exam questions here. It's just, if I stop to do exam questions, I won't get through this topic in the air. I may choose to go through the exam questions in another video, and you could then watch that video and see the answers to these questions. Because the questions, if anything, are more important even than the notes, as it were, in terms of understanding stuff. In terms of obviously doing well, you have to practice questions. But as part of the package, you have all the questions and you have all the answers. And I think the best thing for the answers is that if you want to do a Skype session with me, and I'll Skype you through the answers. And most of the questions you'll be able to sort okay. You'll have the answers with them, so you'll be able to look at the answer and say if you've got the right answer. You'll be able to also look at questions that you couldn't understand why that was the answer, and you can Skype with me about that, and we can talk about that. By the way, this topic's very possible. It looks hard at the start, especially it looks hard for AS students, especially at this time of the year because you're just out of GCSE and possibly miss bits of GCSE as well. So you may feel uncomfortable, but you may think you missed stuff that other students maybe did, but I don't think so. If you did the topic in GCSE biology, double award or triple award, that involved this osmosis and pieces of potato and solutions, if you remember that topic, it also had transpiration in it. If you did that, then you've got the same as everybody else. But so you might still feel uncomfortable. You feel this is a difficult topic. This is a difficult topic. This is a topic everybody's difficult with. And as I said earlier at the start, some A2 students, when they get to A2, they need to know a bit about this, and they've either forgotten it or they never quite got it in the first place. So if you're having a bit of difficulty with it, no problem. It's not easy. It's not that easy. And I find it difficult to explain even because it's still quite difficult to explain. So there we are. That's the first thing. Red blood cells in pure water, red blood cells in strong sugar solutions. What happens to them? If you put a red blood cell in an isotonic solution, that simply means the solution has the same water potential as the red blood cell. Nothing happens. The red blood cell is protected. And so where has this got relevance? Well, in the real world, In the real world, you're not usually putting red blood cells into water, strong sugar solutions. But its relevance with regards to living organisms, little single-celled or more cells, multicellular little organisms that live in water, live in rivers or in seawater. If you're living in a river, the water there might have a water potential close to zero because it's maybe almost pure water. So you may have issues then because your body, your cytoplasm of your cells may be minus 400 water potential. So water's constantly moving into your cells. So you're living in the river. So if water constantly moves into your cells, you might burst. Your cells might burst. But usually the organisms have strategies or ways of getting rid of the extra water. If you live in seawater, then it would be quite useful if your water potential was exactly the same as the seawater, then you have no issues. Water will neither move in or out of your cells. But that's frequently not the case. And once again, organisms have means of dealing with that, and that's something you would look at possibly even next year, but not in this topic, not in great detail. If there are questions do come up on that in the question booklet, then that's the sort of thing I would go through with you in the question booklet. So there we are. We've dealt with solutions and animal cells. Now here's the tricky one, the awkward one. We have to now talk about plant cells. And for plant cells, we've got slightly more complications. We have to get rid of this for a second. With plant cells, things are a wee bit more complicated. It's a wee bit more complicated because with solutions and with animal cells, the formula was the water potential is equal to the solute potential. Okay, and that applied to the solutions and the animal cells. In other words, if you knew the solute potential, then you knew the water potential, and you could just interchange those two terms. You could talk about the water potential of a red blood cell being minus 300, and you could say its solute potential is minus 300, therefore its water potential is minus 300. But when you're dealing with plant cells, the formula has got an extra bit too. It's got another bit here, plus the pressure potential. And this is more often the kind of question you get asked. You get asked about plant cells. Plant cells are more difficult. And you get asked. And this third term here is something called the pressure potential. So the water potential of a plant cell, how do you get the water potential of a plant cell? You need to know its solute potential and its pressure potential. And when you add those two together, you get this value. So to get the water potential of a plant cell, you need to know this and this. Okay, so there's two terms that are involved in determining the water potential of a plant cell. Now, with regards to a plant cell, what do these terms refer to? Well, this term, the solute potential, refers to the contents of the plant cell. It's cytoplasm and what's inside the vacuole. So the liquid that's inside the plant cell. What does this term refer to? This is all to do with the cell wall. Pressure potential, think pressure, think wall. Pressure potential to do with the cell wall. I'm going to try to explain that to you now. I'll go through that and look maybe possibly at a question on plant cells. So once again, this is all spelled out perfectly in the notes here, and all the stuff I'm saying, and hopefully with the notes in front of you, you can understand it. So I've said here, plant cells are slightly more complicated. You need to use this formula for plant cells. Calculate the water potential of a plant cell. You need to know both its solute potential and its pressure potential. The solute potential of plant cells is to do with its cytoplasm and its vacuole. So this term is to do with the liquids that are inside the plant cell. the cytoplasm and vacuole of the plant cell contains dissolved stuff. It's not pure water, it's dissolved solutes, causing the cell to have a negative solute potential. So because the cytoplasm of a plant cell is the same as the cytoplasm essentially of a red blood cell or an animal cell, it's got a negative value. It's got a negative value, so negative solute potential. As I say, the pressure potential is to do with the presence of the cell wall, which is pressing in. Cell wall is pressing in on the outside of the cell, or supporting the outside of the cell. And what this harks back to is, if you go back to GCSE, you might remember that if you put plant cells in pure water, they didn't burst. And they didn't burst because they still expanded, but they didn't burst because they had a cell wall, and the cell wall prevented them bursting. So if a red blood cell had a cell wall, it mightn't burst. But plant cells do have a cell wall, and the cell wall prevents them bursting. Instead of a plant cell bursting when you put it in pure water, if you remember, the plant cell became turgid. It became full up with water and swollen with water, but it didn't burst. And that's where this comes in, this so-called pressure potential. So and we're going to look at, so the bottom page 13 I said, for example, when we place a plant cell is placed, when a plant cell is placed in pure water, water will tend to move into the plant cell from higher to lower water potential. So here's what I'm going to try and explain this, and this it's quite an extended explanation, but hopefully it helps you somewhat. So what we're going to look at first of all is we've got a beaker of water. Again, this is a beaker. So this is pure water. Pure water. Pure water here, and so its water potential would be 0. Okay, and what we're going to do, if we're going to put into the pure water, we're going to put a plant cell. Here goes our plant cell. And we're going to put the plant cell like this. That's meant to be the cell wall of the plant cell. They're around the outside cell wall of the plant cell, like that. And I'm just doing this as an illustration to see if you can see if we can understand this. So there's the cell wall of the plant cell. And then inside the cell wall, I'm going to do this. I'm going to put the cell membrane. And hopefully you can see that there. Well, I'll draw it first of all and you can look. If you can see that there, just about, you can see that the cell membrane is not touching up against the cell wall. I'm going to put the vacuole in the center as well. Small vacuole. So you might remember this kind of plant cell from GCSE. This is a cell that's undergone plasmolysis. In other words, this cell has been shrunk, as it were, by putting it in a strong salt solution. And you can see the cytoplasm's on here, cytoplasm. And that's the vacuole in the center. And you see the cell membrane is not pushing hard up against the cell wall. Okay, so we're going to start with this kind of cell. And we've placed it into pure water, into a beaker of pure water. Now, if we went back to GCSE and I asked you, what do you think happens to that plant cell? You might say, the pure water moves into the plant cell and the plant cell starts to expand and the cell membrane starts to, the vacuole gets bigger and the cytoplasm gets bigger, and eventually the cell membrane pushes up against the cell wall and it pushes and pushes and pushes, and the cell gets a bit bigger and then it stops getting a bit bigger and the cell is said to be turgid. Essentially that's what happens. So that's the GCSE version, is the pure water moves into the plant cell. Now, however, I want to explain it in terms of solute potential and pressure potential. So if I asked you first of all, let's look at the solute potential of the cytoplasm and vacuole of this cell, solute potential. And if I asked you to think of a value for the solute potential of the cytoplasm, of the contents of the cytoplasm vacuole, so you might say, well, I'm going to make up an example. I might say it's minus 800. So the solute potential is minus 800. Why do I say it's minus 800? Because it contains lots of dissolved solutes and so on. So the solute potential of that cytoplasm is minus 800. Now, if the solute potential is minus 800, that's because there's lots of dissolved solute in the cytoplasm and so on. Now, at this stage, notice that the cell wall is not touching up against the cell membrane. So this term is not important at this stage. This term actually has a value of 0. Okay, so this term is a value of 0 because the cell wall is not pushing up against the cell membrane. But the cytoplasm has got a value of minus 800. So if I asked you a question, what do you think will happen to the water in this beaker? And you would say, well, I expect the water to move from the pure water into the cell, from the high water potential here into the cell, which is a low water potential of minus 800. So its solute potential is minus 800, so its water potential is also minus 800 because the pressure potential is 0. And so water moves into that cell. What then happens to the solute potential if water moves into the cell? Well, the solute potential might go from minus 800 to say minus 700. Yeah, because more water is moved into the cytoplasm and the vacuole. It's more dilute, minus 700. Okay, so if I asked you now, what's the water potential of this cell now? And you would say, it's minus 700. So the cytoplasm and vacuole have expanded a little bit, but not too much. And if I ask you then, do you expect water to move into that cell now? You would say, oh yes, because the water potential out here is 0, and here it's minus 700. So you see where this is going. Okay, so gradually more and more water moves into the cytoplasm, and the effect that has is to keep bringing down causing the solute potential to become closer to zero. So it might become minus 600, more water still moves in, minus 500, and so on. But eventually, what happens is the cytoplasmic vacuole expands, the cell wall starts to push up against the cytoplasmic vacuole, or the cytoplasmic vacuole push up against the cell wall. The cell membrane pushes up against the cell wall. And this value here, the pressure potential, starts to take on a value. So let's say water has moved in and the solute potential is now minus 400. So a lot of water has moved in and the vacuole and the cytoplasm will expand. And the solute potential is minus 400. Water will still continue moving in. And this then might come down to minus 300. Okay, so it continues to become more closer to zero, so it might be down to minus 300. But suddenly at this point, what you might discover is that the cell membrane is now pushing against the cell wall a little bit, and the pressure potential comes into play. And all of a sudden the pressure potential becomes plus 100. Okay, so the pressure potential is plus 100, solute potential is minus 300. Okay, and why have we got a pressure potential now? Because the cytoplasm is expanded enough to push against the cell wall. So we've now got a pressure potential. So if I asked you now, what's the water potential of the cell? You would say, oh, it's equal to the solute potential plus the pressure potential, which is equal to minus 300 plus 100. Oh, that's minus 200. Okay, so the water potential of the cell is now minus 200. So the solute potential is minus 300, pressure potential plus 100. So the water potential is minus 300. I'm sorry, the water potential is now minus 200. Water potential of the cell is now minus 200. Water potential outside zero. Will water move into the cell? Water will continue to move into the cell. And maybe what happens is this comes up, so this becomes more negative, let's say to minus 150. So the solute potential might become minus 150. It's getting more and more dilute, minus 150. And what you might discover is the pressure potential has come up to plus 150. So now the solute potential and the pressure potential are exactly equal. What's the water potential? It's equal to the solute potential plus the pressure potential. So it's minus 150 plus 150, which is zero. So the water potential of the cell becomes zero. So the water potential of the cell becomes zero. The water potential outside is also zero. So does water move in or out of the cell? Answer is no. No more water goes into the cell. The cell membrane the cytoplasm vacuole have expanded. They're now pushing up against the cell wall. The cell wall is pushing back against the cytoplasm vacuole with an equal force. So at this stage, the solute potential and the pressure potential are equal and opposite. And so the overall water potential of the cell is zero. The cell's now said to be turgid. No more water moves into it. Does that make sense? So the cell's now got a water potential zero. The water outside is a water potential zero. No more water moves into the cell. The cell is said to be turgid. So this is a you won't get asked to explain this in detail, but it's useful to understand what's going on here. Okay. Now, so that's what happens. You place a plant cell in pure water. Water will either move in or out according to the water potentials. I've asked a question on page 14. What happens if you take one of these turgid plant cells that's got a water potential of zero and you place it into a strong sugar solution? Well, the turgid plant cell will have a water potential of zero. Place it into a strong sugar solution, its water potential may be minus 1,000. Water will be sucked out or pulled out of the plant cell and it'll become plasmonized. So water will be pulled out of the cell and it'll become plasmonized. So we'll go back to this kind of shrunken sort of shape. On page 14 here, I've then shown, it's a very good diagram that looks at how plant cells behave in different solutions then. And you actually need this diagram or a similar diagram to be able to understand from here on in what's happening. So the only thing really that remains, that's the main explanation of water potential and solute potential in animal and plant cells. What remains is possibly look at a couple of experiments, and there are two experiments that are in the specification. One of them is about measuring the water potential of plant cells, and that's essentially, if you go back to GCSE, that was where, you remember GCSE, you cut pieces of potato and you put them into a range of different solutions, and you're looking to see which solution the plant cells neither got bigger or smaller, the potato pieces neither got bigger or smaller, the cells neither increased in size or mass or decreased in size and mass. And you could say then that where the solution in which the potato pieces were placed when there was no change in their size or shape, that solution had to be isotonic with the piece of potato, with the cells of the potato. So you're able to then estimate the water potential of the potato cells. By the way, you don't have to know how to calculate the water potential of a solution. So for example, if you made up a solution in a beaker of water with salt in it, you don't have to be able to calculate what its water potential is. That would normally be got from a textbook. The technician in school would work that out and would be able to figure out how much salt to add to water to give you, for example, a water potential of -700 or whatever. So that's not something you need to calculate. But you definitely need my notes here to be able to look at page 15, for example, and understand what's going on page 15. And it's really, I'm not going to put it up on the screen or anything, it's just too complicated, but it's self-explanatory. Explanation of those experiments. The first experiment to estimate the water potential of potato cells or plant cells, explained there thoroughly, and the second experiment to find the solute potential of plant cells at incipient plasmolysis. Those are two experiments that come up in the experimental exam, the practical exam in June. Okay, and those are experiments you'll be doing in class, writing up in your practical notebook. Okay, I think I'm going to stop there. That took almost an hour, and that's why I'm going so fast. There's a lot of stuff to say and do. But the key to this is to have a set of my notes, because even without this explanation that I'm doing on the board here, you could figure most of this out from the notes here. They're very clear and very precise. And as I say, you send me a text, and I post the package of notes and booklets and everything else to you. It'll arrive the next day, and then you can either watch this video or you can Skype with me. And I assure you, it'll help a lot. So hopefully soon I'll have a video done on the other half of this topic, which is about diffusion, facilitated diffusion, active transport, and endocytosis and exocytosis. An equally important topic. If I'm looking through the question booklet here, probably easily half the questions are from that topic. So you have a sort of almost 50-50 chance in an exam getting a question to do with osmosis, what we've just been doing here, or a question to do with diffusion, facilitated diffusion, etc. And you also have a good chance of getting essays on each of these two topics. And in recent years there's been essays about osmosis and essays on diffusion, facilitated diffusion, etc. And they've been nice essays, actually quite easy essays. So there you are. Thank you very much.